And for quiz, this is 5% of my module, or which can say 5 marks of this module. And so the total you will be given 10 marks, and there would be 6 questions. 10 minutes, and you have six questions. And out of it, basically, I will have only 5 as a best performing. I will count only 5. So this quiz again will be for only 10 minutes. So you are supposed to be in that particular meeting, your respective PA meeting. I will give you basically all the instructions, detailed instructions soon. So at this moment, I'm asking, if you have any questions regarding the quiz, then you can ask me. And after this, once I start the lecture, there won't be access. I won't be able to answer any questions. Okay, Chandan.
Yes, the syllabus will be till today's lecture also, right?
I will announce everything.
Okay.
Any other question? Okay. Fine.
So is it compulsory to attend all 6 questions or?
Only best 5 is going to be counted for this. So you attend 5, you attend 6, doesn't matter. Only 5 will be there. You can attend 3 also. But the other best you will get marks only 3.
Sir, at what time will the quiz be? At the start or at the end?
So you are from day 3, your timing basically, the tutorial time is 2 to 3, right?
Yes,
so exact time start would be 2:05 and it will end on 2:15. So automatically it will be conducted through Moodle.
Yes.
Okay, next one. Mohit. Okay, I'm going to stop here then. If nobody is listening, then I'm going to start the lecture. Khan, you can lower your hand.
Yes, but will it be on Safe or Moodle?
I just announced it's on the Moodle.
Okay, thank you.
Yeah. Okay. I'm going to start the lecture now. So again, this is the last lecture of this module. The next lecture will be conducted by Professor Srilaja Nair. And in this particular lecture, I'm going to cover a lot of information about the Genius part. So these are our, learning objectives. First, we'll talk about inherited DNA leads to specific threads, then DNA packaging, and then how DNA basically, the information on DNA is being converted into protein. So what is this? Exactly central dogma, and in central dogma you have 2 sub processes. You can say transcription and translation. We'll talk about that, and if at all time permits, I will talk about genetic code and mutation. So first, and the question over here is how the information is stored in DNA. So if you look into the DNA structure, it is double helix, and you have the complementary strand. So there is specific, base pair will have, base pair will have specific interaction. For example, A will always have hydrogen bonding with T, C will have hydrogen bonding with this, C will be having hydrogen bonding with G, and so on. So you have DNA, this upper DNA running 5 times to the 3 times this side, and the lower DNA is basically exactly opposite direction. So this double helix DNA has a particular direction. One will be having let's say this directionality is from let's say right to left. Then the complementary direction, complementary strand direction is from left to right. So this is very specific. And when you think about this particular sequence and you think about its partner, and because there will be only one chance, or since you have the A C C A A, you can say this sequence, you can see the complementary sequence as T T G G T. So this is very unique thing. and because of that it can store the information, and that stored information with the help of transcription and translation will get into formation of the protein, and that will be the last part of the lecture, towards the last part of the lecture. Now, when you want to, we have discussed about it last lecture that the DNAs are very specific in terms of copying. So you have these two parental molecule, or you can say two parental strands, and this parental strand will separate into template, and in the next generation of cycle also, what would happen is it would form new strand using the template strand, and the new strand will be complementary to the template strand. But then the question over here, during this process, when the new strands are new strands are basically being created with the help of the template strand, are there any chances that there could be some errors? And if at all there are errors, then what could be the consequences? So the answer to that question is very unique, we can say, because first you have to think about the directionality of the synthesis. So again, when the DNA is being synthesized, it gets synthesized in particular direction, and that particular direction is from 5' end to 3' end of direction. So when you think about this 5' and 3', so you have this deoxyribose sugar, you can say over here, and when you think about the numbering, this is number 2, and at the second position, since there is no oxygen, and that is why it is called as deoxyribonuclease, deoxyribose. And from there you have the DNA aspect, the nomenclature. So there is a specific bonding. G will have bonding with C, C will have bonding with A, and vice versa. And because of that, it's very specific. But still, there are some chances to have the error. And this, when DNA is basically being synthesized, there is a special enzyme called DNA polymerase, which helps the DNA synthesis. And this particular enzyme, DNA polymerase, has, as well as it can synthesize, as well as it has capability to check the errors during the synthesis. And so when you think about this particular DNA polymerase, that particular enzyme can serve as your, if you have to consider an analogy, that can be talked about as a pencil which can write, or in this context it is synthesizing in the 5' 3' direction, and as well as it can delete the basically any errors. If at all there are any errors with the eraser, it can be basically the line, let's say you have drawn something which is unwanted with the eraser, it can be traced. Similarly, DNA polymerase can check the error, and it has capacity to correct that error. So this DNA polymerase, is extremely important in terms of the DNA synthesis. But if at all, let's say hypothetical question over here, if at all there is something goes wrong and still the error correction is not done, then there is a mutation, and this mutation could be either natural, just because DNA polymerase or some other components of this DNA synthesis did not work, and because of that there could be mutation. In such case, the mutation would be natural, and then mutation could be also induced, just like when you when you basically sit for long time under X-ray or under UV rays, then it will change your genetic basically content, because these are high energy radiation, and that can break the bonds. After breaking the bonds, you won't be having that particular interaction, and that's why there are chances to have the mutation. But apart from that, there could be some mutation, even though there are some mutations at few places, but still it can be a silent. It is not expressed, or and with the silent mutation, what will happen is the apparent effect on the organism is very much similar. So these are, the mutation. When you think about mutation, it could be beneficial, as well as it could have a drastic effect. But When you think about the mutants, and there is a general terminology it will come when you do the experiment, and this during the experiment, always you will have a wild type. What does this wild type mean? What does this mutant mean? So in this particular context, the wild type that would be a reference state organism. So the organism which is in, let's say, native state, it has not been changed at all, and that would be your wild type. But in case of mutant, organism might have gone under some changes in the genetic makeup. And genetic makeup in terms of, if you compare that genetic, whatever the sequence with respect to the wild type, then it would be mutant. And the wild type, you can say it has a peculiar trait that are found in a population. Those are found in population. So those are the few normal things which are being found in that particular population. That is called as a wild type. And whatever changes happens within the wild type, whatever the genetic changes happens within the wild type, that would lead to the mutant. So when this terminology comes, wild type and mutant, you have to understand that wild type and mutant, these are two distinct organisms. Then when you think about wild type and mutant, then again, basically these are with respect to the trait or with respect to a particular gene. And the mutant can have one or more genetic changes. It totally depends with reference to the wild type molecule or wild type organism. But now over here, to give you an analogy, I'm just showing you two pictures. On the left, what you see is normal population, and it's within the population you can see there is a lot of diversity, because each individual has its own characteristics, and those will be because of those will be very much relevant when you reach to some you can say younger childhood also. And over right side, what you see is basically series of X-Men, and you have Wolverine, you have Magneto, and other characters are there. And if you have seen these movies of the especially from the X-Men, all these characters are called as mutants. So in nutshell, this is your, on the left side, this is your normal population that can be said as a wild type, and on the right side you have the mutant. Similarly, in this particular picture, which is on your left side, the upper one, what you see is you have the 3 deers, and among 3 deers, one is albino, and the other 2 are basically brown. Those are that is basically a regular female deer. And the lower picture, you have raccoon, and in the raccoon again, the white one is an albino one, and it's a regular color is basically can say brownish also. So there are some changes, there are some mutations, and because of those mutations, sometimes the traits are very evident, sometimes traits are not that evident, and so when traits are not evident, you can say the mutation is silent. When trait is visible, then you can say the mutation is no more silent. Just our case, in this case, this is the color of normal fruit fly, which is red. And in case of the mutant, then you might have seen some fruit fly which has white color eye. So the white color eye is basically the mutation, and it is not seen very commonly. So there is some variation, and in this particular case, the red color fruit fly will fall as a wild type, and the white color eye that fruit fly that will fall as a mutant. Now, when you think about cell division, and last lecture what we learned is there are 2 specific cells in context to that, and each cell will have 2 particular, you can say, pathways to have the production of cells. one is meiosis, other is mitosis, and basis of that we will have the haploid or declared form. But when in terms of reproduction, you see that the general term being used as gametes, or those are cells which are being useful for the reproduction, and the other category is the somatic cells. So somatic cells are again, when you think about it, it is very helpful in terms of maintaining your wear and tear, replacing the few cells which are constantly getting replenished. But in an organism, when the mutation has to be transmitted, that will get transmitted only to the mutations in the germ cell and not in the somatic cell. So then what is exactly this genetic makeup, and how does it basically reveal the particular trait? So based on the genetic makeup, you have a terminology called as genotype. So genotype is a terminology where you can say it's a genetic content of an organism, and on this basis of genetic content, the genotype will express a particular trait, and this particular trait could be, let's say, the color of the organism, hair of the organism, eye color, or something else. And the visible trait, those are visible trait, direct observable trait, and those traits are basically inherited, and that is called as a phenotype. But other question is, how does the information from phenotype to phenotype is being carried, and what is exactly mechanism behind it? And that's kind of a little hard to answer question back in those days, where the scientists just started learning about the gene and all those things. So in that perspective, this particular physician, Dr. Gerard, what he did is he basically identified some disease called as alpha-proton, and this particular alpha-proton is present in everyone's body. And with that alpha-proton, what happens is your urine, it looks black. And to convert that into the yellow color, there is a specific metabolism involved over there, and there is a specific enzyme that is called as alpha-proton. So basically, that particular enzyme will, convert this alpha-proton to the yellow color. And if someone doesn't have that particular enzyme, then the color will remain black, and this could be an inborn error of metabolism. So this kind of, the first finding the term inborn error of metabolism without knowing exactly the situation over there. This term, and with that, one can see that Dr. Gerard was really ahead of his time. And this is in, you can say, late 80s and early 90s. So it's, almost 100 years back, more than 100 years back. So another, some examples, in order to understand genotype to phenotype and all those things, there is one more person whose contribution is exemplary, and that person's name is Gregor Mendel. And in particular, Gregor Mendel's principles of heredity apply to human as well. So he had, very beautiful set of experiments, and those experiments basically gave us idea how the gene is being passed from one generation to other generation. In this particular diagram over here, what you see, the phenylalanine is basically getting converted into acetic acid. So if at all it is not being converted, then you can say it is a disease, and it will require a particular metabolite which has to be there in the body. If at all your body does not have it, then you have that disease. So what one can confirm over here is gene will dictate the production of a particular enzyme, and cell synthesis or degradation of organic molecules uh, can be, that can be done by metabolic pathways, and each chemical reaction involved in this metabolic pathway is catalyzed by particular specific enzyme. So then, the question over here is, what is the relation between the genes and enzyme? How these two are related to each other? And again, back in 1800, you didn't had much means to interpret the data. You don't have much means to collect the data. What data has to be collected, and as such, the concept of gene at that time was evolving. But then, when you think about the particular traits, the genes, which are basically responsible, these are some lists. So there is, gene can be responsible for color code of animal, which is like, it has to change the green, it has to have some kind of stripe or so. So all those things are basically code color. And in particular, it's basically the color of the eye in Drosophila. So white eye is basically a rare genetic, you can say, change, and white eyes in the Drosophila. And then other, basically we talked about this black urine in alpha-protonuria with that particular disease. But then the question is, what exactly these genes do? And in order to understand these genes, and then, there were many experiments were conducted, but the major progress in this kind of, the detecting, or you can say, finding the relationship between the genes and enzyme, that came, with these two gentlemen, and with the George Biddle and Edward Totten. So these two gentlemen basically worked on you can say mold, which is Neurospora crassa, and this is a mold which can be seen now on the bread. And what they did is they took the wild type where nothing is being changed, and they also, so they asked series of questions, and in that basically their approach was little different. And at that time, the biggest question was how the genes are responsible for particular phenotype, and how the particular phenotype can be observed with the genes. Instead of asking this question, what they try to understand is, they wanted to understand what is this gene which controls which reaction. It's a little different approach. And in order to have that answer, what they did is they basically took this Neurospora crassa, and they basically exposed to two kinds of medium. One is minimal medium, other is complete medium. So minimal medium is basically a medium where it has some inorganic salts, glucose, biotin, and you have the agar as a solution support also, or support medium. But then this minimal medium, which will have the only small amount of nutrition, and the rest of the nutrition will be biosynthesized by this Neurospora. The best thing about Neurospora is it requires less amount of you can say molecules to survive. So whatever the biosynthesis or whatever the biosynthesis happens inside the Neurospora, it is not like very expensive, it is very small. And that way it can manage to synthesize few molecules by itself and go for the metabolic activity also. And when you think about the complete medium will consist of everything, or it will have the minimal media, it will have the extra things. The extra things could be amino acids, some few other nutrients, and those nutrients could be iron, which would be important towards the growth. And what they observed that wild type neospora can grow even in minimal media. And there were couple of instances where the minimal with the minimal media this particular plant or this particular fungi did not survive. So what was all those things? Where what they did is they had the wild type plant and they had the mutant plant, and these mutant plants were obtained by exposing it to the X-rays, and these X-rays will change the genetic makeup, and that is how they got their mutant. And after getting the mutant, they isolated this, and then they saw that it is unable to survive in minimal media, which is having very few nutrients, very required, you can say. So this is where workflow. The wild type is there. From wild type, they have the X-ray, and for the X-ray, then they got to see the both ends. In one end, you can see there is a complete. When you have this mutant and you provide the complete medium, there is a growth, and when you have minimal medium, there is no growth. So it's a very interesting observation. And then they did a series of the experiments over there, and in this particular series, in this particular example, what you see is on the picture on the right side, there are two test tubes, and two test tubes. One test tube has the control media, which is minimal media, and other is basically complete media. And what they see is when you put the mutant in the minimal media, then there is no growth, and wild type can have the growth in the minimal media also. So there was an interference that from this experiment that mutant is not able to synthesize one or more specific components, and because in the minimal media there is not much growth of the mutant. So then what they did is they prepared different vials, and different vials has different composition in the minimal media. So just in the first it is minimal media along with component 1, then component 2, and so on. And in some cases you will see they notice there is growth, and in some other cases there is no growth. So then so in particular, they were interested in arginine biosynthesis, and their work was being followed by other two colleagues of them, and they basically invested further. So that their names are Adrian Serve and Norman Horowitz. So what they tried to do is they wanted to see the synthesis of arginine, and this synthesis of arginine, you have different reactions, and at each stage of reaction you can say there is some kind of activity has to be done, and this activity can be governed by particular enzymes. For example, precursor to ornithine, that the reaction is being catalyzed by, let's say, enzyme A. The ornithine to citrulline, that reaction is being catalyzed by enzyme B, and the third one, citrulline to arginine, that reaction is catalyzed by enzyme C, and so on. So this is the you can say the pathway, and when you think about the arginine is one of the 20 amino acids that from there the possible combination from the all possible combinations of amino acids that would have various possibilities, and there would be a polypeptide. Because of the polypeptide, that particular protein can be there. Now coming back to their experiments, and what we see, this is the first column, and in first column they have the wild type, and you have the first row is basically you have the minimal media. Second row you have the minimal media plus one chemical that is called as ornithine. The third row is minimal media with the citrulline, and fourth is minimal media plus arginine. And now when you look into this result, the wild type over here, so no matter it is a minimal media with or without this particular chemical, still with the wild type there is a growth. But when you think about the mutants, class 1, class 2, class 3, at at least one instance you can see there is no growth. So for example, you just have the minimal media and there is no growth in the class 1. But then when you have the minimal media along with ornithine, there is a expression of the mutants. Similarly, over here, whenever you see this yellow band, that means there is a expression. But in the other mutants, what they saw is even they you add some media, still the after adding the media, still there is no growth. Sorry, the minimum media along with the few components. And you see, in particular this column. In this particular column, what you see, the first there is no growth, and the last one there is a growth. So what that confirms, I mean, what is the summary about? With this column, what we can say that it can grow with or without any supplement, this particular wild type. But when it comes to the class 1 mutant, it can grow on ornithine, citrusin and arginine. All three are required, and that's how it can produce the arginine. The second mutant has only citrullin and arginine. So if you have the minimal media plus ornithine, then there is no growth. But then it requires other two components. The third one is it is only seen with the presence of the minimal media along with the this arginine. So what that means that this particular this particular test tube or it will require arginine to grow. No matter without there is, let's say there is no arginine, then there won't be active growth. So with that experiment, we can basically interpret with this that in the case of class 1 mutation, this precursor to ornithine, that particular reaction is being inhibited. In the second case, this ornithine to citrullin, that has been inhibited, and the third one, citrullin to arginine, that has been not happening. So those were overall the experiments, and in that way the people thought how the gene is related and what is exactly their relevance. And when these two gentlemen, when they proposed that gene is indeed genetic material also, at that time this Edward Tatum and George Biddle over there, what they propose that each gene will have the that one gene will correspond for one enzyme. But then with the later on evidences and all those things, it was later confirmed that that's not the case. And from one gene to one enzyme, the theory got modified. And from one gene to current theory is one gene, one polypeptide. So, in particularly, yeah, this is what initially this was the hypothesis: one gene, one protein, or one gene, one enzyme, you can say. And but then when you think about the protein, at the times they have several polypeptides, and each polypeptide will have its own gene. Then that was the case. In that case, the original hypothesis by Biddle and Tatum was changed, and then it was basically said that one gene will have one polypeptide. So when you think about the chromosomes and genes, again the chromosome is nothing but you can say compact form or dense form of the DNA. But when you think about the traits which are being inherited, then it could be related to color, it could be related to shape, it could be related to some kind of size. So it's various things which are related to the genes. So for example, the human eye color, that is you can say that is inherited information, and or it is being expressed in the offspring, and depending upon the color of the parents, that would get decided. And to observe, this basically those those are observable traits. The color, shape, these are peculiar characteristics. And those basically one of the first experiments were done to understand the relation between so-called genes and traits that were done by Gregor Mendel. And then it was followed by, in case of Gregor Mendel, he experimented with the pea plant, and other person, Thomas Morgan, he experimented with the fruit plant. So when we think about the Mendel's contribution, it's a very amazing, whatever he did at that time, it was back in 1800, amazing thing, very amazing thing. So, and then his work was, remained unnoticed for a long time. In this particular experiment, what it is called as law of segregation, and what he did is he took fewer true breeding plants, and in this, one breeding plant has a purple flower color, the other has white flower color, and then he basically went for the crossbreed between them. And in the first generation, that is called as F1 generation, P generation is basically parent generation. F1 generation, that is called as first offspring, which is offspring by the parent generation. And in that particular generation, what you see is you have all plants in the purple. So basically, the white color over there is not seen at all. Then the purple color is over here, and then again he basically went for the self, you can say pollination or self pollination between these purple color flowers plants. And in this next subsequent generation, that is F2 generation or second generation, what he saw, like he had approximately 3 purple flower plants versus 1 white flower plant. That is like kind of intriguing. And then from this statement, what he thought, that there are some heritable factors, and the heritable factors, there are some traits. So and some traits are recessive, that is not being expressed, and some traits are dominant. And if at all there is a combination between dominant and recessive traits, if that is the combination within the gamete, then only only dominant trait is being expressed. So which is very really good observation. And at the time he called these genes, at that time he called them as alleles, alleles, alleles. And what he said is that this alleles will determine the organism's appearance, and the dominant will show up always if at all there is mixture of dominant versus recessive traits or recessive alleles. And the recessive allele will have no significant or you can say it will have no noticeable effect on the organism's appearance. So when you think about this F2 generation, and totally he could see total somewhere about 705 purple flowers plants, and the white were 224. So this particular ratio is 1:3 or 3:2, basically 3 purple flowers, and for each 3 purple flower there will be 1 white flower. This is kind of approximate you can say ratio. And when you want to think about the this particular law of segregation, and when you want to think about the gene, let's say the purple flower gene, and now we will think about the gamete. So here the true breeding plant, let's say the purple one, that has a genotype, let's say that genes or alleles, you can say that is in capital PP, and the white one has small pp, because this is recessive, this is dominant. And this is the way of writing. And then when you think about the gametes will be always haploid. So this is a diploid form, and the gametes will be haploid form. This is what we discussed in the last lecture. And in case of purple flower, this is capital P. In case of white flower, this is lowercase p or small p. And now, when you have the first generation, then what you should have is half of the flowers should have this purple, and half of should be have white. But instead of that, what you see, you have let's say the complete form after the gamete formation and all the things. In this particular F1 generation, you will have this genotype PP. But even though the PP is there, still the color is the capital P and small p. The color is still purple. So in this case, the dominant trait is getting basically expressed, and the recessive trait is not getting expressed. But now, when you think about the F2 generation over there, and over there, what he did is he took the sperm from, you can say, the F1 generation and the egg from the F1. This is called, you can say, self pollination also. And if you think about the arrangement, this particular box or this particular square, it is called as Punnett square, and this Punnett square will show the combinations of all alleles in the offspring. And then then what you want to do is you want to basically you have this particular PP and this PP, and what you do is you just write here, and then you take the combination. So in particular, let's say for this particular element over here in this matrix, in this square, the matrix in this matrix, this particular element, I can write the combination of the capital P over here and capital P from here. The next one, the capital P from here and small p from here. And for this particular element, I will take the capital P from here and small p from here. And in these two cases, capital P and small p, that would have a dominant effect because of the dominant trait will get expressed. And that way, from this particular probability or this particular possible combination, what I can predict that there would be 3 purple flowers and 1 white flower, and that is what exactly Gregor Mendel saw. And in his case, basically, you can think that whatever he did, that was just you can say that was pure experiment, and he did not calculate this probability. But then later on, the other scientists, the Punnett, and that is why these squares are called as Punnett square. He basically tried to make sense in terms of the probabilities and all these things. And with this combination over here, what we can see, the combination of these gametes will result in 3:1 ratio of F2 generation. And this is what exactly the Mendel observed. The next law or next observation of the Mendel was again astonishing. So next law is called as law of independent assortment. And in that particular experiment, what he did is he basically took 2 traits. And here 2 traits over here in this case, what you see is this is the yellow seed which has a round shape. And the other is basically green seed with the wrinkle shape. The gamete of this one, the yellow and brown, that can be written as capital Y, capital R. The gamete of the green and wrinkle, that can be written as small y, small r. And then you basically take this, the cross breeding between these 2 gametes, and the cross breeding should have the expression of y, capital Y, small y, capital R, small r. And in this particular case, since the dominating trait in color, that is gray, that is yellow, and dominating trait is the round. So even though you have the combinations of these things, still you see over here you have the round shape. So in this particular case, the both threads which are being dominant, that is being expressed. Now the question over here is, when we thought, if you have two threads, what should be the lineage look like, or the SU generation look like? And if at all the two threads are independent, then the ratio should be different, phenotypic ratio should be different. Phenotype, you remember, that is the appearance. So if at all, if the hypothesis, let's say the two threads are dependent on each other, then you should have seen the phenotypic ratio of 3 to 1. So in that case, what would have happened is, you have the gametes capital Y, capital R, half of the gametes like that, and half of the gametes will have small y, small r, and over here, and in that case you would have seen this result. So 3 to 1. So the 3 will have yellow and round shape, and 1 will have wrinkle and green color. But then in his case, what he saw, the second generation over here, he saw that there are 315 round and yellow seeds. Then you have green color, green and brown, that is basically 108. Then wrinkle and yellow, those are 101, and green wrinkle, that is both traits are recessive, that is being 32. And now when you want to basically think about this hypothesis as independent, then in that case your sperm gametes will be written like this, all possible combinations over here, and similar for the egg. And now then you want to basically take this combinations of this sperms and gametes in particular row and that particular column. And then over here what you see is, out of basically 9 will, out of 16, basically this is basically probability, and in this particular probability you will see the ratio, phenotypic ratio will be 9:3:3:1, and this the 9 will have yellow and round shape, the 3 will have green and round shape, 3 will have wrinkle shape and yellow color, and 1 will have wrinkle shape and green color. And this is what exactly Mendel saw. And that is why it is basically, what he said is, two traits are not being basically dependent, they are independent. So this is what basically the second law of Mendel states. These two traits are somehow phenotypic traits, they are independent. And now in our the next objective is about DNA packaging and how exactly these things are being accessed in terms of the genes and all these things. It's very much similar to library, and when you think about the library, you have different cells, and different cells will have compartments and all those things. This is what you can think about the chromosome itself or DNA packaging. So again, when you think about the DNA, and just to give you some kind of idea, you can think about DNA and the dimensions. Last lecture we discussed about the dimensions of DNA. So you have 10 base pairs. The 10 base pair distance is 3.4 nanometer, which means one base pair will have distance of 0.34 nanometer. And you remember there are 23 chromosomes in the human, and if you take chromosome number 1, it has approximately 250 million base pairs. And now if I have to calculate how much that length would be, that length would be 250 times 10 to the power 6 times 0.34 nanometer, and that would be roughly 85 millimeter. But here is the thing, 85 millimeter, which is, you can say, 8.5 centimeter, almost like your your small scale. So it is that basically you can say 8.5 centimeter, which is visibly seen. But when you think about the cells always not seen with the naked eye. And when you think about the eukaryotic cell, the distance and the dimension eukaryotic cell will have some micron size. But now you compare, you have the DNA of this length, 8.5 centimeters, inside that micron size cell. How does that happen? Which means, I mean, the DNA has to have some kind of compact form. It has to have, a lot of other things involved in that. Now, when we think about the organization of the DNA into the chromosome, this is how it looks. So, on the first level over here, you have the ribbon model, which is basically a double helical structure, and the width of that double helical structure is 2 nanometers in that. And the base pair between the one helical turn has 10 base pair, and that is basically 3.4 nanometers also. Now, what happens is this DNA, the first packing is due to the basically something called as chromatin, and the first level packing of DNA in the chromatin would be some proteins are involved over there, and those proteins are nothing but histones over here. So these histones will interact, and then you will get this kind of shape. So DNA is getting wrapped around this histones, and histones has some tail, and you can say there is a ball shaped head, and some histones will come together, they will get binded, and along with that you have this DNA wrapped around, and then you have that particular structure. When DNA is bound or wrapped around this system, that is called as nucleosome. And in the second level of packing, so basically this histone will interact and interact with the histone cells also, and then it will make this kind of structure. So it will make this fibrous structure, which will have the width of, you can say, 30 nanometer or so. This is the fiber. You can see first level over here, the DNA is getting wrapped around this histone. Again, the histone is basically, again, you can say it takes some kind of spiral form or file form over here. That makes the fiber, and the width of that fiber could be typically of the order of 30 nanometer. Then further, this histones, you can say, they are coiled in this manner, in this scaffolding manner, and that basically scaffold over here, that width is 300 nanometer. So don't get, these are not actual dimensions over here. The dimension over here you see 30 nanometer, and over here you see 300 nanometer. So that's the actual is basically you are saying the compressed version of this, or you can say from this point you are seeing the magnified version, and from this point you are seeing the magnified form of DNA over here. And then at the end over here, the 300 nanometer fiber, that is basically that is what the chromatin is made for, and this this is the chromosome. These are two system chromatids, and that is how it is packed. But then, if you remember, the chromosome that appears only time of cell division. Otherwise, it doesn't appear, and otherwise, it is in some different form. So, when you think about accessing this DNA, there has to be some kind of potential library, or just like when you want to access some books inside the library, there is some kind of code, and you have to look into that particular code, and then you will go to the particular cells to look for it. So similarly, in case of finding the gene on the chromosome or on the this chromatin, the same similar process you can say, or I'm just giving you analogy. So very similar way, and over there you need to locate. And those locations are very unique. And during the metabolic activity, the genes are basically located, and with that, with the help of those particular genes, particular enzymes, particular protein can be expressed. But when you think about the accessing, the DNA by default will have its very complex, a compact structure, or you can say. And there are the two terminologies over here. When it is accessible, that basically it is called as euchromatin, and in case of euchromatin, the DNA or this particular chromatin is dispersed, less compact, and it will have accessible form. So over here, just you can see this depiction over here. You can see these histones are there, and in between histones there is some distance gap, or gap is there. So those could be basically nothing but you have some some chance for the interaction with the gene, and with that you are able to get the whatever the location you can able to get those proper locations, and then once you get the access to the gene, then particular protein can be expressed. But the other form is heterochromatin, which is basically a very compact form, and in that compact form you cannot have access to the gene. So again, you think about this chromosome, and inside cell, inside cell again there are 23 chromosomes, but these chromosomes are in the pair. So for each chromosome will have its a pair. So there are basically except 23rd, but then 23rd in case of female that's XX chromosome, and in case of male it is XY. And in the cell, all these basically chromosomes are spread out. And when you just want to see, this is basically fluorescence image, and this is arranged, you can say. These are chromosomes are arranged into the karyotype, but inside cell it is very different features. And when cell is undergoing the cell division process, then the chromosome will basically look like this. Otherwise, it is mostly distangled, disarranged, and then you can see a lot of blurry image over here. So this is how the chromosome will look like in the normal cell. And then next we'll talk about this DNA to protein, and we'll talk about the basic principles for the transcription and translation. Now here let's say what we learned so far is there is some, and how exactly that information is being translated into, how exactly that ends up in getting the protein expressed inside the cell. So that is the whole process called as central dogma, and within that you have different sub-processes. The first process over here is you basically get a transcription, and this transcription is nothing but synthesis of RNA molecule using the information from DNA. Now you may ask me, what is RNA? Now RNA and DNA, they are essentially, you can say, very similar molecules. Now in case of RNA, if you remember that sugar base, we talked about this sugar base over here, and this is deoxyribose, okay. And at the second location over here, the 2' location here, oxygen is not there. This is a hydroxyl group over here in the third location. And when at the 2' location there is a hydroxyl group, then that would be a ribose. And from the hydroxyl group, if you don't have that hydroxyl group, you have just hydrogen over there, then it is called as deoxyribose. That is one of the difference. And the other difference over there is you have different purines and pyrimidines. So in case of DNA you have ATCG, and in case of RNA you have AUGC. Again, when you think about this purines and pyrimidines, in case of DNA you have ATCG, and in case of RNA, you have AUGC. Instead of thymine, there would be uracil. These are particular, two differences in case of RNA and DNA. And even RNA will have the backbone of the phosphate molecule. Going back to our point about the transcription. Okay. So that's one of the difference, or you can say two differences. So in first process, when the information from DNA that is being transcribed in terms of RNA, and in this particular case, that RNA is called as messenger RNA. The short form of that is mRNA, messenger RNA, mRNA. And RNA is basically, you can say, the in this case, in this process, RNA is the bridge between the genes and proteins that they encode. Over here, firstly, we are talking about the bacterial cell. And now remember, in the bacterial cell, there is no aspect nucleus. The DNA and all that information just inside the cell, it's in the cytoplasm. There is nothing as such, nuclear compartment or something like that. There is no nuclear envelope over there. And from mRNA, what happens is, further this information is being translated. And this translation, this process, translation, is basically synthesis of polypeptide chain using the information in the mRNA. So you have the mRNA. That mRNA is going to get interacted with the ribosome. And so what happens is, this mRNA gets inside the ribosome, and inside the ribosome you have a different mechanism. Basically on the mRNA you can say there is a codon, and there would be a tRNA which is charged with the charge, means it will have, it's not like electrostatic charge over there, but the tRNA charge, when you say tRNA is charged with the amino acid, that means tRNA will have a particular amino acid at the end of their basic structure. So, and that is what, basically the tRNA will have the anticodon, and that anticodon will read the code, and if anticodon and there is a match between codon and anticodon, then the amino acid which attached to the tRNA, that will basically get into the ribosome, and then the next code comes, next codon comes. Then there would be there has to be proper tRNA coming over there to read that codon, and then what happens is the previous amino acid will get attached to this new coming amino acid, and then there would be a synthesis of polypeptide bond, and such there would be many polypeptide bonds, and with that there would be polypeptide, and this polypeptide is nothing but the initial protein, and this polypeptide will get into the secondary structure and ternary structure and quaternary structure. So there would be different foldings involved over there. Again, the energy principles are used over there. The molecule, when you think about it, it's a big molecule, but then it would get minimized. It would, minimize in terms of the energy, and it would try to go into the lowest form of the energy, and then the protein will form. So that is the process of the translation. And in this bacterial cell, particular bacterial cell, the translation can be concurrent with the transcription. What that means is, in the bacterial cell, the transcription and translation, both processes can go hand in hand, can happen at same time. So this is about the prokaryotic, or this is about the bacterial cell. The process in the eukaryotic cell is little different, and over here what you see is the polyribosome. This is the cartoon depiction, and this is the electron micrograph of how this the ribosomes are interact with the mRNA. So on single strand of mRNA, you see there are multiple ribosomes, and that since you have multiple ribosomes, then it is called as polyribosome. and this is the 5 prime end of the mRNA. This is 3 prime end of the mRNA, and this mRNA has essentially same gene, right? Basically same information, and that information is being decoded by ribosome molecule. And you can see, if at all, on the single chain of mRNA there are different ribosomes. Essentially they will produce same protein, and in this case what you infer is, with this particular single mRNA, at a time there could be 4 proteins making exactly the same protein. The 4 copies of proteins are being made. I mean, this is first, second, third, and fourth. And then once it is the polypeptide is done, then there is a stop codon, and with the stop codon the whole process of polypeptide making that stop, and then the peptide is being released into this cytoplasm, and further process happens. Now at the this end, 5 prime end, the ribosome, the subunits can attach, and then the process can go on. So, and this is what this picture is. You can see this line over here, that is mRNA, and the other things over here, the big things over here, those are nothing but some kind of globular or some kind of circular structure also, that is called as, those are ribosomes. And in this case, in case of bacteria, this, as I said, the transcript and transcription process can go hand in hand. That is what is shown here. So what we show here, see here, this is the the faint line over here, that is nothing but DNA, and that is shown over in the cartoon figure below. It's shown in the yellow, shown in the blue color. And at the these nodes are nothing but RNA polymerase, and that is basically from DNA the RNA mRNA is being synthesized over here. And on each mRNA you see multiple ribosomes, or you can say polyribosomes also, and then that is what exactly seen here. This faint line is nothing but DNA, and the nodes over here, at each node there is a RNA polymerase, and this you see dot, and within that dot you can see a faint line, and that faint line is nothing but your mRNA. And this is this is 5 prime end of the mRNA, this is 3 prime end of the mRNA, and at this you can see many copies of ribosome, many copies of the proteins are being made over here. Now coming back to the transcription in eukaryotic, that is a little different situation over here. And in this particular case, what happens is, because you have the nuclear envelope and the transcription, the process of getting the information from DNA to RNA, that happens inside this nuclear envelope, and there has to be some kind of check and balances, because the mRNA has to come out of this nuclear envelope, and it has to get protected, and that is the process is little different. So that's why you have first pre-mRNA, and then in the second step you have the mRNA, and in this case mRNA is modified through RNA processing to yield finished mRNA, and there is, you can say, there is some kind of protection involved over here. So basically the whole machinery in the eukaryotic cell is a little different than the bacteria. And how exactly these codons are have to be read, the ribosome complexes, they are little different. I don't want to get into that particular detail, but then there is certain differences are there. That's what I want to say. And then the second process is process is translation, and again in translation what happens, whatever the mRNA code is there, that is being basically mRNA is being in interaction with the ribosome, and with that you will get the polypeptide, and then that basically ribosome is in the cytoplasm. Now, you remember in the first, I think second lecture also, what we discussed is there is a nuclear envelope, And just to adjacent to nuclear envelope, you have the endoplasmic reticulum, and you have smooth endoplasmic reticulum. You have rough endoplasmic reticulum. The ribosome will sit on the rough endoplasmic reticulum, and that's where the things are, over there. I mean, you can see those are, basically synthesis sites for the different biomolecules. There could be lipid formation and all those things, which is happening on the smooth ribosome, but in case of rough endoplasmic reticulum, it's basically ribosomes are sitting over there. And in case of eukaryote, you have to understand that the nuclear envelope will separate the transcription process from the translation. That's one of the bigger differences. And when you think about the RNA processing over there, when you think about the the transcription, RNA processing and transcription, over DNA will have many genes, and over here you see one part of the gene, and then you have the exon 1, intron, exon 2, intron, exon 3, and so on. The exon and introns, although it's a part of the DNA, but there are very subtle differences over here, very major differences you can say. Exons, these are basically, you can say, coding regions, and introns, these are non-coding regions, which means whatever there is in exon, that is going to get expressed. So basically, this particular exon that will give me this domain 1, which is shown in the domain 1 of the protein, this particular protein, which is shown in the green color. And then there is the intron. The intron will not have as such effect, but then I think and then there are some regulatory mechanisms, and that's why there is some even though there is no code involved as such, but still that gap is there. And then it comes to exon 2. The exon 2 shown over here is in the orange color, and then there would be this is domain 2, and there is basically with the exons you have the this attachment from domain 1 and domain 2 when it gets expressed, and similarly exon 3, which is shown in the purple. So the introns, they as such they won't contribute in terms of the protein formation. That particular gene is not going to be useful. That particular part of the gene, but exons, that particular part of gene is being expressed in terms of protein formation or polypeptide formation. And this, I mean, this is little. Again, when you have, when we think about the bacterial cell versus prokaryotic cell versus eukaryotic cell, you can say, both cell will have its functionality, but then in case of eukaryotic, you have kind of compartmentalization, and because of that, these regulatory mechanisms could be coming. And overall, when you think about Vishal, I will, I'm just going to finish the lecture within a couple of minutes, and then I can answer your question. When you think about the, overall the central dogma, this is what the flow looks like. From DNA, the information is being transcribed, or that process called transcription. From DNA, it has become, RNA, and from RNA to protein, that information is being transmitted, and the process is called transmission. But this central dogma theory, initially it was like this. Basically, it's a concept that basically tells you about the cell, how the cellular chain of command has to be followed. And regularly you will see, in prokaryotic and eukaryotic cell, there could be regular mechanism from DNA to RNA, that is transcription process, and from RNA to protein, that is translation process. But then when you think about the viruses viruses as such, you have virus particle. As such, it is not you can say dead or alive. But then when virus comes in contact with the host cell, then it can get activated. And typically, these viruses may have the RNA as their genetic material inside their capsule. And this, the viruses has a different process. What they do is they use the host mechanism, and in order to use that host mechanism, this RNA which is being uh, uh, in the virus, that first will get converted into DNA. That process called as reverse transcription. And then with that process, when it synthesizes the DNA, then it will basically act, then it will have transcription process and then transcription process. And that is why, if at all you get infection, viral infection, you will see, you will have, at a time you might feel low, you will have high fever also, because what is happening is essentially your, the viruses, those are being, they are using your all metabolic activity, and they are using that metabolic activity to replicate themselves, and then your body is resisting. There are lot of antibody mechanisms are involved. So overall, you need lot of you can say metabolic activities, and you might be, because of unnecessary developments happening because of this viruses, you are deficient of energy currency also, and that's where you feel low, you feel less energetic, and all those things. So yeah, so basically this is called as from RNA to DNA. That is very special case, that is called as reverse transcription. And so basically this is and the last part is genetic code and mutation, which I will try to finish in the couple of minutes. I don't have much time. But in case of DNA, you think about you have A, C, G, T. These are 4 letters, 4 characters, or the 4 bases are there. But in case of protein, there are 20 letters, or you can say there are 20 bases, and these 20 bases are nothing but your amino acids. And you can have the genetic code possibly in terms of all these 4 or 20 letters. The question was whether the genetic code is being passed from the DNA or protein, and initial guess was it is protein, but then basically with experiments, series of experiments, and when people basically got to know more about the DNA, then it was confirmed that DNA is indeed the genetic material. And in the basically transcription process, you have this mRNA, and mRNA has this codon, and each codon basically is made of 3 nucleotides. UGG. This UGG will have the amino acid, particular amino acid expressed as cysteine, and then the UGU that will have the phenylalanine. Then GGC, you have the glycine, and so on. So basically, there is a possible combination, and then some basically the particular over here the code AUG, that is very specific, and that is for the methionine. That is the starting of the protein synthesis. And every protein synthesis, no matter it is eukaryotic or prokaryotic, you will see the starting codon is AUG. And in case of stop codon, when you have to basically stop the protein synthesis, that is basically you have these 3 codons: UAA, UAG, and UGA. And in between, you might have different codons, and depending upon that, you have different depending upon what is the combination and all those things, there would be typical expression. So in this case, you have the UUU and UUC. Both codons will have phenylalanine as an amino acid, and UUA and UUG will have leucine. So there is a particular chart which is given in the book. You can have a look later on, and this is basically, when you think about that, that is figure 17.5. You can have a look over there. What is basically the particular code, and for that particular code, what would be the amino acid, and that is mentioned over there. But interesting thing, let's say if at all there is a mutation. So this is the 3rd position over here. And if at all in the 3rd position over here, that is called oval position. And this is this particular uracil is being mutated to cytosine also. And in that case, the codon will be UUC, but then still it is expressing phenylalanine. And then in this case, the mutation is called as silent. As such, there is no difference in the amino acid. So that's why the mutation is being called as silent. But then when you have the mutation in the 1st codon over here from let's say UUU to GUU, then in that case it will have a value and there is basically missense. I mean there is value in place of single mutation. This particular mutation will be a missense mutation. And why sometimes it's very important because let's say in wild type hemoglobin you have the cell shape something like round, and in sickle cell hemoglobin you have the cell shape which is some kind of you can say this this shape. But in this mutant the sickle cell is nothing but there is a mutation at particular location. So in this, the 5' location over here, you have GSG, and in that basically, if you have the GUG, and in that GUG case, what happens is you have this valine. Instead of glutamine, now in sickle cell, you have the valine, and that changes this, surface properties of the sickle cell, red blood cell in this case, and that that would result in this kind of shape of this RBC. And then with this reduction, then it has less capacity to carry the oxygen, and then person will feel anemic and all that. So that is then you have the nonsense mutation, and then the nonsense basically protein synthesis stops. Where this protein synthesis stops, that is termed as that mutation is termed as nonsense. So UUA2 and then UAA. These are basically, you have the UAA, that is the stop codon. So mutation, this particular mutation will, UUA, that is for the leucine, and now if it is mutated to UAA, then it will have the stop codon and stop in between. So this is called as nonsense. And now I think, I just stop here, and now I can take the question. Yes, any questions? Yes.
Sir, if AUG always has to be present at the start of the
yes.
DNA chain, does that mean all
proteins? No, not AUG on DNA. AUG, that is on the mRNA. AUG, this is on mRNA. Okay? Not on DNA. DNA has a different code. This is the DNA code over here. And ATG will have AUG. EAC will have AUG. Okay. And it will be basically, it has to have, so in order to have, how does the protein synthesis will start? You have, lot of code on the mRNA. And the mRNA code doesn't start just with methionine. There is, before methionine, there could be, and that is where I didn't explain it little bit. Those are, certain differences in case of bacteria and the eukaryotic. So there is, there are specific sites where the mRNA sits inside the ribosome, and this site has to be recognized by the ribosome, and that is where the subtle difference is there. And what would happen is inside the ribosome, once before this AUG, there would be a lot of other codons. But then when this AUG particularly comes in that zone, you can say, zone in the 30th ribosome or small subunit, then and then the process of protein synthesis can start. So every time the first amino acid, no matter it is prokaryotic or eukaryotic, the first amino acid would be methionine. Okay. Any question? Mantara?
Yes, sir. So all protein chains will start with methionine.
Yes, by default.
Okay.
Yeah. Any more questions? Yes. Nice.
Sir, I wanted to ask regarding the sickle cell anemia.
Mm-hmm.
Sir, In that the coding will change in each and every cell or in just a single cell?
Coding will change in each and every cell. Yes.
That means how will it happen, sir?
So the thing is that now you think about person. Yeah. Every person has this DNA. That mutation is happen in the DNA itself. Okay, it is not on the mRNA level. So that the mutation is on the DNA level itself, and because of that, the person by default, you have that I don't know where exactly which chromosome this gene is located, but then you know if it is on the mutation is on the DNA level, it is basically individual that is being faced with that disease, and these are some kind of hereditary diseases.
So like they are transmitted from the main DNA chain only, right?
Yes, but then yeah, I mean see, the thing is that at a time what would happen is how the crossing over. I mean, remember last time when we talked about this meiosis and mitosis. So meiosis has this crossing over, and during that, I think the first thing is that either parents should have. I mean, one of the parents should have sickle cell. Now I don't remember whether it is a male parent or female parent. I don't remember that. I think that will be taught in the in the your tutorial session. But then this is most commonly inherited disorder, disease in the Africa, and I think in India there are, I think, part of India, probably in the Nagpur region also, we have a lot of sickle cell patients. So this is I mean again, I don't remember whether it is from the male or female or maybe both parents. I mean, it is basically yeah, I mean, and there are different types of sickle cell. So if you have to in case of homozygote and in case of heterozygote, let's say heterozygote may show some symptoms, but they might not have a particular trait. So many things are there which I don't remember at this point.
Okay, sir. Thank you, sir. Yeah. Thank you, sir.
Okay. Any other question? Okay. If there are no more questions, then we can stop here. And just remember that on your tutorial session, next tutorial session, there would be a quiz. Thank you. And from next week, Professor Srilaj and I will teach next module of the MCB. Thank you.